welcome to episode 15 of the BioLogic podcast. Today I'm going to be continuing our mini-series on genetics by exploring the structure and function of our DNA, namely the vital processes of transcription and translation. In the previous couple episodes, I introduced the basics of genetics with Mendel's famous pea plant experiments, and I also discussed the process of meiosis. I feel like I've done a lot of talking about DNA without really ever going into detail on just what exactly DNA is, or how it's structured, or how it works. Well, all that is going to change today, as I dive into the immensely rich field of study that surrounds deoxyribonucleic acid. Today you will learn about how DNA carries genes, and the biochemical mechanisms through which genes are expressed as proteins. If you listened to episode 4 on nucleic acids, you should be familiar with the basic structure of DNA, but for the sake of simplicity, I'm going to briefly recap some of the major information. Nucleic acids, like DNA, are biopolymers, and these are composed of monomers called nucleotides. Each nucleotide is a ribose sugar attached to a phosphate group and a nitrogenous base. The phosphate group and the sugar are used in the backbone of DNA, while the nitrogenous base is used in coding information. There are 4 different nitrogenous bases in DNA: adenine, guanine, cytosine, and thymine, also called A, G, C, and T. When you have 2 complementary strands of DNA, the A and the T, or the adenine and thymine, they can pair together with 2 hydrogen bonds, and the C and G, the cytosine and guanine, can pair together with 3 hydrogen bonds. When 2 complementary strands bind together, all of their base sequences, the C's and G's and the A's and T's, they're all complementary, and so they line up and they all bind together, and it creates like a zipper between the 2 DNA strands, and it stabilizes the molecule. One of the reasons DNA is such a great molecule for information storage, for heredity and that kind of thing, is because it's very stable, and all of these hydrogen bonds contribute to its stability. The structure of DNA also gives it a directionality, and it possesses a hydroxyl end and a phosphate end. Respectively, these are called a 3' end and a 5' end. And this is because the 3' end has a hydroxyl group on the 3' carbon, and the 5' end has a phosphate group on the 5' carbon. DNA is synthesized only by adding bases to the 3' end, but I'll be getting to DNA synthesis and repair more in the next episode. In order for DNA to work, it must be expressed. So what does it mean for DNA to be expressed? As DNA has 4 different nucleotide bases, these are used as a code for specific amino acids, which then form specific proteins. A sequence of DNA bases that code for a protein is called a gene. A chromosome is a single DNA molecule, which is basically a long, extremely thin string, and the sequences along this string contain many thousands of genes. It's very important to understand that not all genes are protein coding. Many are regulatory, involved in coordinating and timing the expression of a gene, while others seemingly do nothing or have a purpose that is not yet understood. As with DNA synthesis and repair, I'll be getting to the regulation of gene expression in a later episode. Today is about the mechanism by which a gene is expressed. This is to say, the way that a protein is made from the nucleotide sequence in a gene. This process has 3 parts, which are called in sequential order: transcription, RNA processing, and translation. Chances are you've heard of transcription and translation. This is the process through which a gene is read by a group of proteins to produce a complementary copy of mRNA, which is then read by another group of proteins to build new proteins. The rate of some of the steps in this process can be startlingly fast, like the transcription factor proteins that move along DNA at 50 base pairs a second to produce the intermediate mRNA molecule. With that summary, I think we can jump right into the meat and potatoes of the topic. I'll start with bacterial transcription and then discuss eukaryotes. Transcription in bacteria begins when a suite of proteins, namely RNA polymerase, its subunit sigma, and some associated proteins called general transcription factors, bind to the DNA and open up a sequence that's about 14 base pairs long. This small sequence contains a promoter region, called a promoter, a sequence that attracts the protein sigma and the RNA polymerase, and this initiates transcription. RNA polymerase is indiscriminate on its own. It'll attach to any sequence of DNA that it's exposed to. But when RNA polymerase is in a complex with the protein sigma, the whole complex is much more selective to the DNA that it binds to. There are different kinds of sigma protein, and different kinds seek out different promoter sequences. Cells in different tissues use different sigma proteins to access genes encrypted by different types of promoters. Bacterial cells can use different types of promoters to access different sets of genes or different groups of genes. The bacterial cell can produce a single sigma protein, and the sigma protein can go on to facilitate the expression of a large group of bacterial genes. Once the RNA polymerase sigma complex binds to the exposed patch of DNA, it proceeds to unzip the DNA strands by breaking apart the hydrogen bonds that hold the complementary nucleotide bases together. On the DNA, the RNA polymerase moves in a 5' to 3' direction. That is, it moves towards the 3' end of the molecule and away from the 5' end. Sequences or base pairs are said to be downstream of a given point when they're closer to the 3' end. Those base pairs or sequences that are said to be upstream of a given point exist closer to the 5' end. So if we say that the first base pair in a bacterial gene is +1, or just at the +1 position, then the promoters in the bacterial DNA are somewhere around -10 and -35. That is to say, there are promoters about 10 and then 35 base pairs upstream from the first base pair transcribed in a gene. In bacteria, these promoter regions are called the -10 and -35 boxes. The -10 box contains repeating thymine and adenine pairs, while the -35 box contains a TTGACA sequence, which is common in a wide range of organisms. Keep in mind that the similar biochemical processes used to replicate and express DNA are fundamental to life on Earth. They're so fundamental that virtually all life possesses them. This touches on evolution, which I'll get into in more detail in the next miniseries. Anyways, sigma is the first part of the enzyme complex to touch the DNA, and it binds between the -10 and -35 boxes. The complex opens the DNA so that both strands are accessible, and transcription begins at the +1 site. So the DNA is being funneled into this protein complex through the RNA polymerase molecule, and as the DNA is being shuffled through the inner corridors of this protein, it's also being opened up. One strand of the exposed DNA is called a template strand because it's the strand being read by the RNA polymerase. This template strand is being used to create a complementary strand of mRNA, or messenger RNA. The other strand of DNA, the strand not being read by RNA polymerase, is called the non-template strand. It's also called the coding strand because it contains the literal bases to code for the protein. The complementary mRNA molecule being constructed is identical to the coding DNA strand, with the exception of having the nucleotide uracil in the place of thymine. If I haven't said this before, all RNA molecules use 3 of the 4 nucleotide bases in DNA. It uses a nucleotide called uracil instead of thymine, and it pairs to adenine just like thymine does. When everything is situated and transcription begins, the sigma protein detaches, and this ends the initiation stage. The second stage of bacterial transcription is called elongation. The emergent mRNA molecule grows by 50 nucleotide bases a second as the RNA polymerase reads through the template strand. New nucleotides are added to the 3' end of the mRNA molecule, causing its 5' end to protrude out from the RNA polymerase. As the DNA is being read, it's being moved through the RNA polymerase, guided by various flaps and bumps and channels on the inside of the molecule. As the DNA helix enters the protein, a protrusion called a zipper unzips the DNA, and both strands are guided through their respective corridors by a second protrusion called a rudder, where they're eventually rezipped or resealed, or they have their hydrogen bonds reformed. The DNA leaves the RNA polymerase molecule just as it entered, in the form of a helix. In other parts of the molecule, the growing mRNA strand is being guided through and out of the RNA polymerase. An intake cavity sucks up a stream of loose nucleotide bases, and these are incorporated into the mRNA strand. Suffice to say that the RNA polymerase is in full action during the elongation phase. DNA is moving through it at 50 bases a second, growing the mRNA molecule at the same rate, and everything is going along grand, until of course it all stops. The elongation stage ends and the termination stage begins when the RNA polymerase reads a stop signal in the DNA. In bacterial RNA, this stop signal in the DNA produces a sequence in the mRNA which folds back on itself to create a hairpin loop. Just like when you take a string and fold it back to run parallel with itself, you'll end up with a little half-circle loop at the end. In the mRNA, this hairpin loop physically interferes with the RNA polymerase molecule, and it bumps it off of the DNA strand. Through this physical interference, the hairpin loop terminates transcription. Transcription in eukaryotes has many things in common with transcription in bacteria. After all, it's a very highly conserved system, genetically speaking, so it makes sense that even distantly related species would have many similarities. As there are many similarities and relatively few differences, instead of going over the whole process again, I'll discuss the differences. So first, eukaryotes have a greater variety in their promoter sequences. This increased number of promoter sequences allows the cell to be far more selective when expressing genes. At the -30 position, or about 30 base pairs upstream from the transcription start site, eukaryotes typically have a region called the TATA box, or a TA-TA box, which is literally a sequence of repeating adenine and thymine bases, or A's and T's. Second, eukaryotes use a series of proteins called basal transcription factors in place of the protein sigma. These basal transcription factors self-assemble into a protein complex at the promoter region, and this helps to attract the RNA polymerase. This naturally introduces a third key difference. Eukaryotes don't use a single type of RNA polymerase. Instead, they use 3 different kinds of RNA polymerase, conveniently named RNA polymerase 1, 2, and 3. This allows eukaryotes another level of specificity when expressing genes. RNA polymerase 2, for example, exclusively reads protein-coding genes. The fourth major difference between eukaryotic and bacterial transcription has to do with the termination stage. In bacteria, a hairpin loop disrupts transcription. In eukaryotes, the RNA polymerase reads a DNA sequence full of adenine, or A bases. This is called a polyadenylation signal, also called a poly-A signal. Immediately after the signal is transcribed into the mRNA molecule, an enzyme comes in and snips the mRNA right before the poly-A signal transcription sequence. This creates a free-floating, newly made mRNA molecule. The RNA polymerase then continues bumping along the poly-A signal until it reaches the end of the gene and falls off the DNA. Fifth, bacterial mRNA can be translated almost immediately after being transcribed. In eukaryotes, the RNA polymerase creates something called a primary transcript, or in the specific case of protein-coding genes, a pre-mRNA molecule. This primary transcript molecule has to go through a series of reactions before it becomes an active mRNA molecule able to be translated into a protein. These series of reactions must be performed because of the nature of the eukaryotic DNA. You see, in bacteria, the genes are continuous. An RNA polymerase can hop on a strand of DNA and print out a functional mRNA, no problem. But that's not the case in eukaryotes. Eukaryote genes are split up by numerous little interruptions, sometimes hundreds, sometimes thousands. These interruptions are just sequences of normal nucleotide bases, but they don't really code for anything useful or functional. When the DNA is transcribed, these interrupting sequences are also transcribed and incorporated into the primary transcript molecule. These interrupting sequences are called introns, as opposed to the sequences that actually code for functional stuff, which are called exons. So the eukaryote DNA has alternating intron and exon sequences, which are transcribed into the primary transcript molecule. To remove the introns and bind together the remaining exons, the cell performs something called RNA splicing. RNA splicing is very complicated, so I'll try to be as simple as I can. It's a process that requires 2 types of molecules. Some RNA molecules called small nuclear RNAs, or snRNAs, and some proteins. Together, these molecules form what are called small nuclear ribonucleoproteins. The acronym for the small nuclear ribonucleoproteins is snRNPs, which we can say phonetically as SNRPs. So RNA splicing requires a suite of SNRPs, and these find and bind the adenine inside and near the edges of the intron, and they also bind to the 5' carbon between the guanine and uracil at the boundary of the intron and the exon. Here, the SNRPs form a superstructure called a spliceosome, which is typically a large and very complex mass of interacting and interlocking proteins and RNA. The SNRP at the GU interface and the SNRP at the adenine base are basically on opposite sides of the intron. When the SNRPs all associate together on the spliceosome, the intron is pinched into a loop with a little tail called a lariat created by the excess RNA in the intron running past the adenine base. The SNRPs cut the guanine and uracil bases, and this breaks the GU interface. The SNRPs then drag the uracil on the cut end of the intron to the adenine base near the other end of the intron and bind them together. The intron is now in a full loop, like a lasso, with the circle forming between the uracil and the adenine, and the tail of the lasso like the little lariat. The guanine on the end of the first exon is brought over to the second exon still attached to the lariat tail. The lariat is cut and the exons have their ends attached. The intron is discarded with the spliceosome, and the new mRNA molecule is a pure strand of exons. I'll discuss splicing a bit more when talking about gene expression, where the cell splices in different ways to produce different mRNA from the same gene. As you might have figured, different mRNA codes for different proteins, which means that one gene has the potential to code for multiple different proteins. RNA created by RNA polymerase 1 and 3 would be ready for translation immediately after splicing is complete. RNA polymerase 2, however, creates RNA that has to be processed further. Protein-coding genes create primary transcription products that are spliced into molecules called pre-mRNA. This pre-mRNA molecule is given an altered guanine molecule, and this altered guanine is kind of stuck on top of it like a hat. The guanine possesses 3 phosphate groups, and has a core structure altered to that of 7-methylguanylate. This chemical hat is positioned on the 5' end of the pre-mRNA molecule, like a seal or a cap, and is thus called the 5' cap. Furthermore, the 3' end of the pre-mRNA molecule is capped off with a tail of adenine bases, and this can range from anywhere from 100 to 250 bases long. This string of adenines is called a poly-A tail, and it's not encoded in the DNA itself. The poly-A tail is added by enzymes after transcription. Both the cap and the tail are used to protect the mRNA from degradation, preserving their function and accuracy. So all of this, from the splicing to the 5' cap and the poly-A tail, are all part of RNA processing, which is the second step in the whole process. You often hear of transcription and translation without mention of RNA processing in the middle, but this is likely because RNA processing only occurs in eukaryotes. It's relatively brief, it's really complicated, and it takes place in the nucleus, in the shadow of transcription. Anyways, this takes us to the final step of the process, to translation, where the mRNA is read by ribosomes to synthesize the amino acid chains that will fold into functional proteins. So just like transcription, translation has 3 parts with the same names. Initiation, where the process begins, elongation, where the product molecule grows, and termination, where the process is halted or interrupted. The ribosome is another kind of ribonucleoprotein. Its 2 subunits are composed of both RNA and protein molecules. These 2 subunits bind together, but each has a different role. The small subunit binds to the beginning of the mRNA molecule produced by the RNA polymerase. The large subunits bind to the incoming tRNA and amino acids that will be used to make the protein. I'll discuss the tRNAs in a moment, but first let me clarify something about transcription and translation. In eukaryotes, transcription takes place in the nucleus. The processed mRNA is shuttled out of the nucleus and into the cytoplasm of the cell, where translation occurs. Bacteria don't have a nucleus, so their transcription and translation can happen in the same place, even concurrently with the same molecules. One can use an electron microscope to see the strands of bacterial DNA dotted with numerous RNA polymerases moving down its length. From these RNA polymerases, you can see the mRNA being printed out in long strands, which get longer and longer as its RNA polymerase moves down the DNA. Numerous black clumps cling to this mRNA. These are the ribosomes, and they print the amino acid chains that fold into proteins. In bacteria, every step of this can be seen happening simultaneously. In eukaryotes, this process is discontinuous, as the mRNA must go through a brief period of travel to get out of the nucleus in between transcription and translation. Okay, so what about those tRNAs I mentioned earlier? What are those things? The T in tRNA stands for transfer. tRNA is thus a type of RNA molecule that transfers stuff. In this case, the tRNA molecule transfers the amino acids from solution into the growing polypeptide chain, or the growing protein. These tRNA molecules are tiny, only 75 to 85 nucleotides long, with an L-shaped tertiary structure, and a secondary structure marked by beams of double-stranded RNA and loops of single-stranded RNA. It just so happens that the structure of tRNAs gives them an amazingly crucial ability. A single-stranded loop in the tRNA molecule exposes 3 nucleotide bases called an anticodon, and a CCA sequence on the 3' end offers a site of binding for amino acids. So on one end, the tRNA can hold an amino acid molecule, and on the other end, the tRNA is presenting an anticodon in the form of 3 exposed nucleotide bases. The L shape of the tRNA is useful in that it provides a necessary distance for the amino acids to be properly manipulated by the RNA polymerase. To attach an amino acid to a tRNA, the cell uses an enzyme called amino-acyl tRNA synthetase. This enzyme catalyzes the phosphorylation event in which the amino acid is covalently bonded to the tRNA. There are 20 different amino acids, and each one has its own special amino-acyl tRNA synthetase molecule that catalyzes its binding to the tRNA. When an amino acid is bound to a tRNA, the product molecule is called an amino-acyl tRNA. Sequential amino acids are bonded in the ribosome to produce a single polypeptide chain. So that's the amino acid, but what about the anticodon? What's the deal with that? What is that thing? You might be thinking, if these 3 nucleotides on the tRNA are called an anticodon, is there a regular codon? This is a good line of thought, because the answer is yes, there is. A codon is a 3-base sequence on the mRNA. The anticodon is a complementary 3-base sequence on the tRNA. When the mRNA is being ran through a ribosome, the ribosome lines up the tRNA such that these complementary codon and anticodon sequences bind together. In this way, the codons in mRNA match specific anticodons in the tRNA, which are associated with a specific amino acid. In this way, the codons in the mRNA dictate the amino acid sequence in the polypeptide chain. But it's not quite so simple. Consider the numbers involved here. There's 20 amino acids, but there's 61 different codons. How does this work? This implies that each amino acid can be associated with multiple codons. But how does this work? Each codon is composed of 3 nucleotide bases. The first 2 bases are very important, as these determine the identity of most of the amino acids. But the 3rd base can afford a little variability. When positioned on the ribosome, the 3rd base doesn't necessarily need to fit perfectly. The 3rd base doesn't really have to follow strict AT and GC pairing. It can be a little wobbly, so to speak, and still work fine. In this way, 61 different codons can be used to code for 20 amino acids, with each amino acid having, on average, about 3 different codons. I keep talking about how these molecules are positioned inside the ribosome, so maybe I should talk more about the structure and function of the ribosome itself. The small subunit has a molecule of RNA, which binds complementary to the mRNA, specifically to a start sequence near the 5' end of the mRNA. This start sequence is AUG, which stands for the adenine uracil guanine sequence that codes for the amino acid methionine. As a result, the vast majority of polypeptide chains begin with the methionine amino acid. The AUG sequence is typically 5 or 6 bases downstream from the ribosome binding site. After the mRNA binds to the ribosomal RNA in the small subunit, the methionine-carrying amino-acyl tRNA then binds to the AUG codon in the mRNA. So we have an mRNA molecule threaded over a small subunit of ribosome, with a tRNA molecule bound to the AUG codon on the mRNA, standing straight up the middle of the small subunit like a toothpick sticking out of the bottom of a hamburger bun. It's only after this structure is complete that the large subunit of the ribosome comes and binds to the small subunit, and this completely envelops the attached tRNA molecule. This ends the initiation stage and begins elongation. I should mention that the ribosome has 5 major interior compartments. 2 are like grooves or tunnels. One of these tunnel compartments is the space between the small and large subunits. The mRNA runs through this region as it's read by the ribosome. The other tunnel region is at the top of the large subunit. Here, the amino acids are placed sequentially, bonded together into a polypeptide chain, and scooted out of the ribosome. The other 3 compartments sit inside the large unit of the ribosome, connecting the upper and lower tunnels with chambers that hold the incoming tRNA. These 3 chambers are called the A site, the P site, and the E site. When the large subunit comes down and binds with the small subunit, the tRNA on the start codon fits right into the P site, right in the middle, with its methionine amino acid positioned in the upper tunnel. During the actual process of translation, however, the tRNAs enter the A site first, like access, A for access. The tRNAs come through and they enter the A site first. An incoming amino-acyl tRNA enters the A site through diffusion, where its anticodon, if compatible, binds with the associated codon on the mRNA. If it's incompatible, it'll bounce off and float away. But if it's compatible, the tRNA is held in the A site, where the amino acids are subsequently bound together. This process causes the bond between the original tRNA in the P site and its methionine amino acid to break. The methionine amino acid now shares a peptide bond with the amino acid of the tRNA in the A site. This second amino acid, however, keeps its bond to its tRNA molecule. A group of proteins called elongation factors then help the ribosome to move each tRNA molecule down one position. The P site tRNA goes to the E site, and the A site tRNA goes to the P site. So at this point, the E and the P sites are filled, the A site is empty, and we have a polypeptide chain that's just 2 monomers long. This process of sliding the tRNAs down the line is called translocation. These steps repeat as the chain grows. A third aminoacyl tRNA enters the empty A site. If complementary, its anticodon binds with the next codon on the mRNA. Its amino acid forms a peptide bond with the amino acid in the P site. This peptide bond formation again breaks the bond between the amino acid and the tRNA in the P site. Translocation occurs again. The original tRNA, the one that got pushed into the E site, is now ejected from the ribosome. The tRNA on the P site, freshly detached from its amino acid, is then shuffled into the E site. The newcomer, the tRNA in the A site, who just added its amino acid to the polypeptide, is shuffled to the P site. This process continues again and again, adding a new amino acid each time. The growing polypeptide chain is directed out of the ribosome, where it begins to fold into its functional shape. Translation is terminated through the use of one of 3 different codons, called stop codons, which are UAA, UAG, and UGA. These are stop codons because there aren't any aminoacyl tRNAs that can bind with them, and when they show up in the mRNA, there isn't an associated amino acid that can continue the polypeptide chain, so the whole process stops. As a tRNA can't fill the A site if it can't bind to the codon, a protein called a release factor steps in to fill the gap. By filling the A site, the release factor is able to break the bond between the tRNA and the P site and its amino acid. This action frees the polypeptide chain, which slides out of the ribosome completely to form a fully fledged protein. When the RNA has been read, the entire ribosomal complex dissolves, and all the subunits dissociate and return to the cytosol. The entire process isn't quite finished yet. After translation, there's a few minor modifications that need to happen. As yet another example of the brilliant naming skills of biologists, these are called post-translational modifications. Most generally, polypeptide chains need to be folded properly to ensure the synthesis of a functional protein. A misfolded protein doesn't work, and in the case of prions, may actively cause damage. A group of proteins called molecular chaperones assists the polypeptide chain, making sure everything is oriented and positioned properly so that the folding goes down without accident or error. This is a largely mechanical process that involves physically repositioning and moving the polypeptide chain. These molecular chaperones accelerate and supplement the natural organizational structure that emerges due to the polarity and relative proximity of the amino acid side chains. These newly constructed proteins can also be given chemical tags, typically small sugar or lipid molecules. These tags serve as markers for other biomolecules in the cell, telling them where this protein is supposed to go, or what it's supposed to do. Other tags are integral to the protein structure itself, and are necessary for its proper function. Another method of post-translational modification involves the addition or subtraction of a phosphate group from the protein. If you listen to episode 3 about proteins, you should have a general understanding of this point. Phosphate groups are very electronegative, carrying multiple negative charges. This very strong polarity causes the phosphate group to react with the polar and charged side chains in the protein's amino acids. This interaction generally causes a significant conformational change in the protein. As form follows function, the conformational change is necessary to activate or inactivate a protein. The regulation of proteins is often dependent on their coordinated phosphorylation or dephosphorylation, which coordinates the total protein activity of the cell. Alright, well, that's about it for transcription and translation. To finish the episode, let me summarize the process again. Transcription occurs when RNA polymerase works with other proteins, like the subunit sigma in bacteria and basal transcription factors in eukaryotes, and that they work together to read the DNA and synthesize a strand of RNA. In bacteria, this mRNA can undergo immediate translation. In eukaryotes, the RNA must be modified with a 5' cap and a poly-A tail before being moved out of the nucleus and into the cytoplasm. Translation begins in the cytoplasm when the mRNA molecule binds with a small ribosomal subunit and an aminoacyl tRNA with a methionine amino acid. After these associate, the large subunit attaches to the complex and begins processing tRNA molecules. Incoming tRNA fit into the A site, where their amino acid is bound to the other amino acids present in the ribosome. Then all the present tRNAs are pushed down a space through translocation, and a new tRNA comes into the A site. Each tRNA molecule loses the bond to its amino acid at the P site and gets discarded at the E site. This process repeats, with each set of 3 bases on the mRNA coding for one amino acid to be added to the polypeptide chain. Eventually, all the amino acids are in place and the polypeptide is finished. A protein called a release factor fills the gap provided by a stop codon and terminates translation. A new protein has been produced from a gene sequence. And with that, I think this episode is over. This was an information-dense episode. If you need to listen to it again to soak in all the details, I think that's a good idea. I hope you learned something cool. Thanks for listening. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogicPodcast. To buy something from the store, go to redbubble.com and search for BioLogic. Or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/BiologicPodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.